Steve Brown, Executive Director, the Peanut Foundation USA, and the Dr. Anthony Whitbread, Research Program Director, Innovation Systems, ICRISAT, India. Please join me in welcoming them.
Good morning. Thank you, Anu. I'd like to introduce the first speaker as Dr. Victor Sadras, coming from the South Australian Research and Development Institute in Adelaide. Victor's worked for many years for SARDI in South Australia, very much looking at water deficit, extreme temperatures, nutrient deficit, and a whole range of other factors in mainly the wheat systems of Australia, Argentina, Spain, and China. And he's also co-editor of Field Crops Research. Welcome, Victor.
Good morning. Our ability to generate data is much greater than our ability to make sense out of data. This gap is particularly important in areas where technology moves fast. And say we've been doing yield maps for many years and we're not getting the full value of that information. The bottleneck, I suggest, is conceptual tools and strong theory. That's my interest, and that's the theme of today's paper. I try to place these conceptual tools in context. Farmers do have a set of goals, and they are very variable as farmers are theirs. For sake of argument's sake, a grower seeks profit in a context of risk. She's got a menu of varieties and practices to achieve those goals. And the farmer organizes varieties and practices in certain arrangements in space and time. In jargon, that's cropping system design. This means breeding and agronomy are at the forefront of technology with a direct impact on farming. And all the rest, physiology, genetics, entomology, is one step removed from having an impact. That's why it's so important for us to link with breeding and agronomy. We depend and we generate data and concepts. That's what I would like to talk about, these concepts. I will sample 3 topics today. First, I'll have a look at yield from perspective of evolution and levels of organization. 2 samples of environmental insights showing that how the crop thermal biology affects drought patterns, and a case of root redundancy in Australian wheat that's explained by finer aspects of the environment. And I'll close with two examples of how we use in phenotypic plasticity as a concept. If we define yield in units of mass per area, we're talking of a population. And this means it's important to understand how plants interact with each other. That we need to look at the principles of population ecology. One of the most fundamental is separating features of the system that are density dependent or otherwise. So nature is structured in a nested hierarchy of levels of organization. This nesting means one level contains the other. We scale down to find explanations. If I'm interested in crop water uptake, crop level, I might need to go down to the organ level, look at roots, or even further down. If I'm working on cell membranes, it's okay and I can stay there. But if I want to claim agronomic relevance, I need to scale up. How far up? It depends on the system and the questions. For example, for orchards, where plants are spaced, maybe scaling to the individual plant is enough. We've seen some examples in vineyards this week. If I'm working with wheat or cotton, I must scale to the population because the interactions between plants are important. If I'm interested in hydrology, I need to scale up further to the landscape. With Richard Richards, we classified traits in 3 classes: the traits that scale, the traits that don't scale, and the traits where scaling is conditional to my experimental protocol. For example, gas exchange. If If I measure stomatal conductance for 10 seconds and want to know what the impact of that is on the canopy, there are well-established rules for scaling, and Elias Ferreres updated those scaling rules for gas exchange in the previous interdrought. So BT is a prime example of a trait at scale. Genes that are expressed where and when it matters, proteins that are functional and reduce the fitness of insect populations, and a reduction in the use of insecticide at the ecosystem level. So BT makes sense from gene to ecosystem. And that's the reason why biotechnology has been so successful in crop protection. Now, if we want to use the same approach for yield, of course we fail. And the common explanation is that yield is too complex. But I don't think that explanation is helpful because I'm blaming yield, and there's nothing I can do about the complexity of yield. I think it's more helpful to say my approach is not good to capture the complexity in yield, so we can do something about my approach. So crop yield, the definition of crop yield has evolved. Before being growers, we were hunters and gatherers, so the energy ratio was the measure of yield like any other animal. How much energy I need to invest to get the food and how much energy I had from food. Then we started to grow crops, and for this farmer, the decision was for this amount of seed, how much I use to feed my family and how much I save for the next crop. So the definition of yield for most of the history of agriculture has been grains per grain, the ability of the plant to multiply grains. In a context of unlimited land, this selected for very tall, tillering, aggressive plants. It's only very recently, a few centuries in this long-term scale, that we started to first recognize land as a scarce resource with economic value. So we shift to the definition of tons per hectare. And that shifts the selection pressure to Donald's less competitive ideal type. So it's a fundamental change in the selective pressure, and we move now into a population trait. Overlapping with this definition that remains relevant, we are moving into a definition that accounts for time, kilos per hectare per year, to account for intensity of cropping. In many systems, we can sacrifice the yield of an individual crop to feed more crops in a season with a net benefit. So this is a summary of the ways to improve productivity at the farm level. We can focus on the crop, genetically improving potential, agronomically closing yield gaps, and at the system level as well, improving the potential at the system level through design, arrangement of crops and activities in space and time, and closing the yield gap. We just published a first attempt to try to define and design a conceptual model to deal with the potential and yield gaps at the system level. So this is an example, and I will not go much further into the system level, but this is an example of a trade-off. Each point here is a county in Kentucky, and the most productive counties are the ones where the yield of soybean is increasing more slowly. And the reason is growers are not that much interested in growing better soybean, but in feeding more with soybean double cropping. So because that has a net benefit. The reduction in the yield of individual crop is more than compensated by two crops. There are reductions in fixed costs, and there is an improvement in the capture of resources, water and radiation in particular. Okay, if we start with this idea that yield is a population level trait, I will show two examples of recent work highlighting these interactions. The first is this. Antonio Holt discovered this pattern in sunflower stands at high density. One plant bended one way, and the next plant in the row bended the opposite way. Plants are virtually avoiding each other. This is a density-dependent process. You can see that the fraction of red and green plants, which indicate whether they are bending one way or the other, they increase with increasing plant density. And we can see through time, it's a highly dynamic process. Plants, seedlings start to bend. That wave progresses, is fixed mid-season, and remain fixed until maturity. So to investigate these patterns, they've done two things. One is experiments, and we've done some modeling as well. The experiments are of two kinds. These ones where the movement of plants was monitored and the microclimate, in particular the light microclimate, was monitored. Another set of experiments with light filters. Jorge Casali is the expert in this area, so he threw the kitchen sink to this and to show that these waves of inclination are mediated by changes in the spectral composition of light as perceived by phytochrome. So we went further to test, because this seemed to me like self-organization in the sense that this is different to sun tracking. For sun tracking, it is the sun. It's an external driver of plant movement. Here, we could model the state of the plant with local rules. The state of a plant in day 2 depended on the state of the plant in the previous day and the state of the 2 neighboring plants. So we could reproduce the density-dependent feature of this system and the dynamics. So this is a very elegant experiment by Monica. The final question, what does it mean in terms of production? So she compared the yield of plants that were bending naturally with those that were forced to remain vertical. So what we have is this bit of mouthful idea, but it's a light-mediated self-organization process that increases yield. So these are interactions between plants in action. Clearly, we cannot capture this in any setting, but a population setting. The second study has been done by Lachlan Lake, who's around somewhere here. He tested Donald hypothesis in chickpeas. This idea of smiling genotypes is an Australian breeder's take on Donald's hypothesis. The smile of the crop is the difference in yield between the border and the middle row. It's not high, but yield. So if the smile is large, the genotype is expected to be low yielding. So phenotypically, we showed that this set of chickpeas fits to Donald's expectation. High yield is associated with a low response to competition. We want genotypes that don't smile. Those are the high yielding ones. Yong Li went to interrogate the genome. He used FST to characterize the genetic background of the two traits, and they don't match. I think that's an important insight. What drives genetically the yield of a plant in a crop and what drives the yield of a set of plants under relaxed competition, the plants in the border have more water, nutrients, light. They are in a different light environment. The genetics of those two traits are different. Okay, to conclude this section, say I have a handful of F2 seeds and I can afford a single row, or I decide for whatever reason that the pot experiment suits me. I think it's useful to ask this question: what set of traits we can measure in that non-crop setting that would be relevant, that would scale? And to have some indication of how traits scale or otherwise, I think it's useful to ask whether the traits are density dependent or otherwise. The environment is a large component of the variation in yield, and we've seen many excellent studies where we are using a more and more refined approach to the environment, and we are, those approaches are returning quite deep insights. But we also see many superficial treatments of the environments. When we split the phenotypic components of the variance of many crop traits, but including yield, E is large, is the dominant source of variation. In a typical experiment of comparison of varieties, If we have current varieties and the ones in the pipeline, we might have 5% difference. Temp would be huge, but the variation in the environment is manifold. Two exceptions to this. One is truly high heritability traits. Seed size might have a heritability of 60, 70%, so G is the dominant component. The second exception is, I guess, more interesting, is my bias. If I screen 200 lines in a handful of environments that are similar, I'll be overestimating G and underestimating E. As a sample, this is a special issue of crop science on G by E. There were many good studies dealing with the environment, but half of the papers still stop at location and season to describe the environment. It's a qualitative description that doesn't provide much information. And in particular, because the tools are there, in particular for drought. Jordan and Miller already drew patterns of stress to show that you need to match the traits and the management to the environment. And Karin Chenou's done a nice review of this. Cobb Chapman developed this approach, where the first step is to model the water balance daily on a phenological scale, then cluster analysis to reduce that to somewhere between 3, 5, 6 type of droughts, and then an agronomic check to verify that this is relevant agronomically. Then we can do a number of things, map the probabilities of the different types of stress, translate location season into drought types. This is the pattern of drought for wheat in Australia, where we've been talking of terminal drought for a long time. We've seen some colleagues in Chile also talk of terminal drought, and I'd say this is not a helpful description because it means different things for different people. And in the case of Australia, terminal drought is placing an undue emphasis on grain filling. If we look at the environment 3 and 4, drought starts stem elongation or so. So by the time the crop is filling grain, yield already collapsed because grain set has failed. It would probably take a generation or more to shift that concept of terminal drought into these quantitative things, and it's very entrenched. These patterns depend on three things: the soil, the climate, and the sowing to harvest window. So if I now do this for other crops in the same cropping window, I would expect same kind of patterns, and that's what we get for pilpea. But we were surprised for chickpea, not finding pre-flowering stress. We first thought this might be related to the model. We couldn't fault the model, so we started to look at why chickpea is different. This is Shalal Abo's account of the early days of agriculture in the Fertile Crescent, and you can see chickpea standing out. It started late, and then these growers have some problem. It took them 3,000 years to fix. And that problem was ascochyta. Again, the difference between plants and population. The individual white plant might be damaged by disease, but it survives, produces seed. But when they started to grow these plants in stands, they were devastated. That's why they gave up. And the solution was to grow chickpeas in summer with stored soil water. And the consequences of that are two and very important. One is, of all this crop set, chickpea is the only one with no vernalization requirement. And the second consequence is that the thermal biology of the crop has been shifted. You would expect chickpea to be more like pea because it's from the same place of origin, but it's a lot more like soybean. So what happens now when we go and grow chickpea back in winter, it grows slowly compared to pea or wheat. So for the same pattern of rainfall, a dry spell between two events, a large field pea crop will be stressed, but a thin chickpea crop would be less likely to be stressed. So here, what we are suggesting is that the thermal biology of the crop is modulating the patterns of drought. We've been working with this set of wheat varieties for a few years now, and the question is how selection for yield has shifted the phenotype of wheat. This is one of the more recent studies. What we found is that selection for yield has trimmed the root system. Root biomass, root density have declined all through these 50 years with no impact on water uptake, at least at the crop level, and with an increase in nitrogen uptake that's from the compensation where roots have a much larger nitrogen uptake per unit root. We can explain this in a number of things. It might be that the conductivity for water has increased, but the parsimonious explanation is John Pasuras. He anticipated root redundancy. To understand why in a dry environment roots might be redundant, we need to look at another set of aspects of the environment. First is the seasonality. The vectors in that map show the intensity of seasonality, and where the vector points is the peak of seasonal rainfall. So in the southeast, where these varieties were selected for, we have winter rainfall. So there's no summer rainfall during fallow the soils don't store water. So if we look at the frequency distribution of plant soil water in the southeast, most of the time we grow crops with the dry profile or a little bit wet, but never, it's very unlikely to have a full profile. The second feature is the size of events in the next map. Red is large events, green, blue is small events. So we start with the dry soil, it rains during the season, but when it rains, it's small events. So we have water from the top to the bottom. We never get a full profile. It's very rare. And when we have more water at depth, what we have is soils with boron, alkalinity, and so forth. So the point is, if there's no water in the profile, there's no point in having a profuse and deep root system. So breeding for yield selected for this adaptation. The carbon from roots that were not useful to get water is moving up to increase crop yield. We've been talking about phenotypic plasticity for a while. When we started, we had some comments from breeders in particular, and the comment was, well, we've been working on this and we call this stability. If a crop is stable for a trait, then we say it's the plasticity is low. So the point was, I was just playing with words, and my point is no, because bringing plasticity to the table, what we are bringing is 100 years at least of science from other branches of biology where this concept has been used to understand phenotypes and GVIII. So this is how we're using this concept to look at the performance of crops in two extremes, in a stressful situation that's drought most of the time and the potential. We are interested in both. We've seen early this week by Peter Carberry the trajectories of profit for growers. They have a few in Australia and other dry environments, few seasons where they make a fair amount of money and many seasons where they have losses. So for growers, it's important to capture the good seasons as well as to buffer the poor seasons. So for this case of Philippine Australia, this the variety with the highest plasticity is the winner. It has the highest potential and the best drought adaptation. So that's clear that that's the winner. And that goes to the concept of stability as well. We normally talk, we want yield stability, but we need to ask where is the stability coming from. In this case, the best variety is the less stable one. This is a case where we want stability because it relates to drought adaptation and good potential. And this one is the one where we illustrate the trade-off. We can go for a variety that's good for potential, but at the expense of drought adaptation. And to make a decision, if I'm a grower and I have a choice between these varieties, to make a decision, we need to go back to our patterns of stress and maps. What's the probability of having one type of stress or the other? The last example of how we're using this idea of plasticity is this set of experiments where we screened chickpeas for adaptation to both water and heat stress. We had a factorial with early sowing and irrigation to measure potential. Then we have water deficit and early sowing and late sowing plus minus water. So we have all the combinations. With late sowing, we've seen how we use late sowing as a surrogate to screen for heat stress. One of the problems is that late sowing had two main effects. One is with warm temperatures, phenology goes faster, so we have a shrinking of the critical periods. And that's most of the time the main factor that reduces yield. And the second factor is untimely heat at key reproductive stages. And it's important to make a difference between the two. So I will not show it here, but we've developed a framework to separate the effect of increasing temperature between 25 and 27 that will reduce yield because of phenological shrinking and truly heat stress. So here we measure number of carbon, nitrogen, and water-related traits. I'll illustrate that with seed number, which is the main yield component. And we have 8 environments. These are the upper and lower percentiles to represent potential and stress. And what we have is here is the varieties with higher plasticity have both high yield potential and stress adaptation. To look at the genetics of these, we can do a number of things. One is the sequential, trying to look at the QTLs or whatever in each of the 8 environments. We have our QTLs and then deal with G by E. The other approach is what Anthony Bradshaw suggests in his quite insightful paper. We need to think 1965 on the state of genetics in those days. He says plasticity has its own genetic control. So instead of asking what's the genetic control of seed number at each environment, we go straight and we ask what's the genetic control of the plasticity of seed number and carbon discrimination and so forth. So we preempt G by E, because what we want for that case is clear. We want high plasticity of seed number. I have time, but I will leave it here.